have become solutions to the world's most pressing concerns. Innovation and creativity are the sparks that ignite to transform lives and society, and these need space to grow.
We are home to these creative and innovative sparks. Our work goes beyond the disciplines, across our 9 faculties, 7 campuses, and being one of Africa's top business schools. As an African global university, we stand proudly among the world's top universities. We remain at the forefront of universal discoveries and provide authentically African solutions to the world's greatest challenges and opportunities. We are pioneers in our fields, from the humanities to cutting-edge technologies, continually seeking ways to improve the world around us. Our hybrid learning systems provide quality education that enables graduates to become adaptable game changers. We have molded nearly 500,000 alumni who have made their mark locally and internationally, and we continue to inspire and equip our students to uphold our firmly rooted culture of excellence. At the heart of what we do is our people. They are creative, critical thinkers who can shape society for the better. Because that is how we teach, learn, innovate, impact, and live. We are the University of Pretoria.
It's a very short video because it's not selling us very well. As the minister is here, he take the opportunity to show them who we are. But anyway, good evening, everyone. Welcome to this great University of Pretoria, at our Future Africa platform or campus. My name is Themba Musia. I'm one of the Deputy Vice Chancellors here. I'm going to keep my remarks very short so that we can enable our expert lecture and other formal introductions to take much of the time. But this is our 37th expert lecture at the university. And those of you who have attended them before know that these are real experts who we select. Is very rigorous on topical issues that change the lives of people and the planet. We're delighted to have the minister here, Minister Steenhuisen, and your entourage. The Vice Chancellor will do other formal introductions, I say. But this topic, and has attracted many people to come here from one of our centers, the Heinz Hoyensen Research Center, where Professor Bastos is the director. We go back a long way at this university in different ways, and I'm truly delighted to be the program director for her expert lecture today. I am now going to introduce the Vice Chancellor briefly and ask him to come and do other formalities before the expert lecture starts. We know how emotive this foot and mouth has become. We know how the minister has been hammered from other quarters on this, and we are very happy that he's here to listen to some of the experts over and above what he listened to over time. The Vice Chancellor, Professor Francis Peterson, is an engineer by profession, so he's going to learn a lot about foot and mouth Yes, I do. And he has been leading this university since October of 24, right? And we are delighted to have him here address us. He's also the chairperson of our University South Africa. It's a forum of all vice-chancellors. He's leading that. He's in his second term. That attests to his deep understanding of higher education matters, because you can hardly lead that body if you don't know what you're saying. We as academics, Minister, have got a lot of egos. Our egos are fat. So if you say something, you better know what you're saying. So Prof. Peterson, this is your time. Yeah.
Thank you, Professor Musia, and Honourable Minister John Steenhuisen, the Minister of Agriculture of the Republic of South Africa. We are indeed privileged to have you here with us this evening. I know that you've got a very, very busy schedule, and I can just indicate to you that we're not going to grill you this evening, but welcome. It's always great to have you at the University of Pretoria. Then members of the Department of Agriculture that are present here, we are very delighted to have you joining us. Then Professor Bastos and the family, her husband and son, who are here in person. I don't know where they are. Are they here? Oh, they're at the back. Yeah, welcome, especially welcome to both of you. And I know that your daughter can't be here, but she's online, so I also want to welcome her. And then to all of you, distinguished guests, colleagues of the University of Pretoria, and also friends of the university. As Professor Musia has indicated, that is our 37th lecture in the UP Expert Lecture Series, and this is a platform that showcases outstanding research at the University of Pretoria. But it also plays another role. It fosters the engagement with the broader public. So it's an expert lecture, and I haven't briefed Armanda beforehand, so I hope that she will present it that way as well. It's an expert lecture, but it's also a lecture that will bring the expert research in a way that the public understands it. And this is one of our ways of making sure that the universities aren't only those institutions on the hill that produce highly intellectual work and highly intellectual output, but people don't, the ordinary public don't necessarily understand it. And our role, Minister, as a university, is also to ask the question, to what extent do our teaching and learning that we do, and to what extent do our research and innovation that we also practice impact society positively? And to do that, we're not working for society, we're working with them. And it's important that the language doesn't nearly disconnect from one another. And it's important that at a public lecture such as this one, the UP Expert Lecture, is also trying to achieve that. So on the one hand, it's a brag session to indicate the quality of the research that we're doing, but in the other way, it's also an educational one to be able to say, this is the value, and this is where the value can be practiced. So this evening's lecture focuses on foot and mouth disease, a highly significant livestock disease with far-reaching economic, social, and regulatory impacts, particularly within South Africa, but also the broader sub-Saharan region. And the lecture will explore the challenges associated with the South African territories, type of viruses, the recent shifts in outbreak patterns that we experienced, and the urgent need for not only transdisciplinary, but also transboundary approaches to disease control. And for me, it's indeed an honor to welcome Professor Armanda Bastos, who bring both scholarly rigor and real-world relevance to this important topic and will be sharing her insights in tonight's lecture. Professor Bastos is the director of the Hans Hohuijsen Research Center and is a professor in the Department of Veterinary Tropical Diseases here in the University of Pretoria's Faculty of Veterinary Science. She obtained a BSc in Genetics in Zoology, an MSc in Genetics, and a PhD in Molecular Virology, all the degrees from the University of Pretoria. And she has supervised or co-supervised the research projects of over 84 postgraduates to completion, and that include 23 at doctoral level and also 38 at master's level. And I hope there are some of your current master's or postgraduate students here this evening to also listen to this expert lecture. To date, she has published 136 scientific journal articles and 7 book chapters, with a primary focus on the molecular epidemiology and diagnosis of vector-borne, zoonotic, and transboundary animal diseases. Her early career research, while employed as a veterinary researcher at the Agriculture Research Council Transboundary Animal Diseases Laboratory, involved typing field and outbreak strains of foot and mouth diseases and African swine fever viruses. In addition to establishing extensive reference sequence databases for foot and mouth disease and also for African swine fever, she developed molecular methods for the rapid detection of these diseases. The PCR detection, and that's the methodology, and the P72 genotyping methods, published in 2003, remain global first steps in molecular diagnosis and typing of African swine fever. Professor Bastos has served in a number of senior management positions, including Deputy and Acting Director of the Mammal Research Institute, as well as Head of the Department of Zoology and Entomology, and currently she is the Director, as I have indicated, of the Hans Hohuijsen Research Center. She was inducted as a Fellow of the Royal Society of South Africa in 2019 and is a member of the Academy of Science of South Africa in 2023 and holds a B2 rating from the National Research Foundation. Her achievements have been recognized by the University of Pretoria through Exceptional Academic Achiever Awards in both the Young Academic and the Senior Academic categories. I trust that you, as the audience, and hopefully those people that are online, will find this lecture thought-provoking and also very enriching. And I encourage you to engage deeply in the discussion that follows. So there will be an opportunity to ask Armanda some questions afterwards. So, ladies and gentlemen, please join me in welcoming Professor Armanda Bastos to the stage.
Good evening, everyone. I see a lot of familiar faces and some unfamiliar faces, and I really look forward to the opportunity to tell you a bit about what has become all-consuming for the last 7 to 8 months, to the extent that I wake up thinking my first waking thought is foot and mouth, with apologies to my husband. So this evening I will be telling you a bit about the research, but also just really setting the scene for what we know about foot and mouth disease and some of the research and regulatory impacts that come with foot and mouth disease outbreaks. So just a little bit about the history of the virus, and it was in fact the first filterable animal virus that was discovered shortly after the first plant virus was discovered. And so this virus is actually very well known to us. We've been studying it for more than 120 odd years. And initially, it was believed that there was just a single virus type. But by the 1920s, 3 of the European serotypes were recognized, A, O, and C. And then about 2 decades later, the SAT types were discovered at Pirbright in the UK. Those are the SAT types 1, 2, and 3. And then finally in the 1950s, early 1950s, Asia 1 was discovered. So there are 7 distinct serotypes of foot and mouth disease, and there is no cross-protection between those different serotypes. So this is really, really important when it comes to controlling the disease through vaccination. The reason why foot and mouth disease is such an important economic disease, despite having a relatively low mortality rate, is because it's highly infectious, particularly in cattle. So the threshold for establishing an infection is incredibly low in cattle. If we compare that to pigs, much higher doses are required to establish an infection by the aerosol route. And it particularly hits high production systems very, very hard. So if we look specifically at the dairy system and the costs associated with an FMD case in a dairy cow, the estimates between the loss of productivity and treatment in veterinary field treatments sit at about 5,000 Rand per animal. So if you add up all of the dairy cattle that have actually been affected to date, you can see that the impact economically is astronomical. And also, because it is such a highly infectious disease, it is a notifiable disease, meaning that you are legally obligated to report it if you suspect that there is FMD. And it is also a controlled disease, meaning that the government and the state actually decides how we control it. And there are very strict legal requirements associated with the control of the disease, both in terms of managing it and when you have outbreaks. And this is guided very much by international regulations and by WOHA, which is the World Organization for Animal Health. But we also have an Animal Diseases Act that is applicable. So there are huge negative trade implications as well as economic implications the moment there is an outbreak. And because of that regulatory requirement, you have to inform the WOHA, which means all your trade partners know when you have an FMD outbreak. There is almost immediate repercussions for reporting. But that reporting is critical because it ensures transparency, which is a core component of the international body regulating this disease. It also has far-reaching consequences that sometimes you may not anticipate, including job losses, insolvencies, but particularly in South Africa, where our levels of food security in 2024, October 2024, were breaching 63%. You can imagine that when there are foot and mouth disease outbreaks and those regulatory requirements kick in, that it has a substantial impact. So the World Organization for Animal Health has codes and manuals which guide how we deal with foot and mouth disease and other controlled diseases. And it also has a reporting system which requires that we notify the moment we have confirmed cases of foot and mouth disease. And this is core to the transparency and governance component that the WOHA framework works on. It's important to mention that these international regulations guide how we deal with the disease in South Africa. And the standards that we use when it comes to controlling the disease are updated in line with new scientific information and technological advances. So the idea here with the terrestrial code is that it should reflect all new scientific advances, which when correctly applied will protect animal health and veterinary public health, whilst avoiding unjustified sanitary barriers to trade. And that last part is really, really important, particularly given the topic of the talk today. I do need to tell you a little bit about the virus, and probably one of the best definitions I've ever heard for a virus is that it's a piece of bad news wrapped in a protein coat. And I think FMD virus definitely meets that requirement. So it belongs to the picornavirus family. That family takes its name from pico, referring to the small genome size, and it also being an RNA virus. And the genome is actually a single-stranded RNA genome, really, really short, just 8,000 nucleotides in length, but can wreak untold havoc. The other important thing about foot and mouth disease is that once that virus binds to a receptor and it basically gets into the cell and starts replicating, you only need a single intact fragment of that RNA to establish an infection. So that some of the earlier work that was done, they took intact RNA. If you inject it into a cell, that RNA will start replicating and forming virus capsids, and it is in fact infectious. And that's also a really important component to keep in mind. So I have to say that the style of lecturing is completely against the way I usually do it. I move around a lot, I use my hands, and I usually use my laptop with a printer, so now I'm going to have to turn away from you, so I'm sorry about that. But essentially, and this doesn't seem to be working anyway, if you look at the structure on the bottom, what's really important about the genome is that there are three distinct segments, the P1, P2, and P3. P1 is significant because it encodes the virus proteins 1, 2, 3, and 4. And 1 to 3 are on the surface of the virus capsid. So this is what is interacting with the immune system when that virus is circulating and replicating. The other P2 and P3 encode non-structural proteins, and of diagnostic significance is a 3D region which codes for the RNA replicase gene. And when we do the diagnostics, we target a really, really tiny part of that gene to confirm FMD presence there. And the diagnostic method we use picks up all 7 serotypes. The other important thing about foot and mouth and where we are in the world is that the SAT types, the 3 SAT types that occur in Southern Africa and in East Africa, if we look at the sequencing and we reconstruct the evolutionary history, they are completely distinct from the Eurasian serotypes A, O, C, and Asia 1. And it's believed that the original virus that led to all of the current serotypes is actually a SAT type or progenitor. I have already mentioned that if there is infection or vaccination against one particular serotype, it does not provide protection against a different serotype. There are some differences, but there are also a lot of commonalities between the different serotypes of foot and mouth disease virus. All of these viruses are acid labile. So essentially, once that pH drops to 6 or very close to 6, the virus just disintegrates and breaks open. And it essentially inactivates the virus, and we use this particular phenomenon when we are trying to inactivate viruses. It is also thermolabile, and this has implications for vaccines and cold chain management. And it's UV sensitive, which is really great, considering where we are. If you especially compare us to the UK, we have lots of sun, we have dry weather, relative humidity tends to be low, but the moment you get to places like the UK where you have high relative humidity and you have overcast conditions, that virus, once it's aerosolized, can in fact survive a good deal better than what it does in our climes. The Eurasian strains also, if we think back to those two evolutionary lineages, we tend to think of them as livestock viruses. So these are maintained within livestock. They tend to spread very quickly. The clinical signs are also quite obvious compared to the SAT types, or at least historically. And because it's associated with livestock and because of the impact on livestock, the focus there is very much on eradication and sometimes historically on mass culling campaigns, as took place in the UK. The SAT types, by comparison, these are distributed throughout sub-Saharan Africa, and the SAT 1 and 2 types have the broadest distribution. SAT 3 tends to be far more restricted to Southern Africa and parts of East Africa, but they are associated with African buffalo maintenance hosts. And generally what we find is that when you get infections in cattle, the clinical signs are not as overt as what you see with the Eurasian serotypes. So also if you're doing clinical surveillance like we do at dip tanks for communities that are living alongside the Kruger National Park where we have FMD-infected buffalo, sometimes it is possible to miss the fact that an animal might be infected because of the mild clinical signs. We also generally, with SAT types, it's associated with lower mortality rates and usually in young animals. And then in terms of thermomobility, meaning its ability to withstand heating up, the SAT types are very, very sensitive to any fluctuations in temperature. So they break open quite easily. They are also genetically and antigenically heterogeneous. So we've always believed, based on this long history of studying these viruses, that the SAT serotypes of FMD are different. And really, for the most part, they are. But what we've seen in the last while suggests that there may be something different going on. We have a very proud and long history of actually controlling this disease very, very well. And we did it through fencing, separating livestock and wildlife, through vaccination of cattle living alongside those buffalo maintenance hosts in the Kruger National Park, and then also through movement control. So people who happen to live alongside Kruger and next to populations of FMD-infected buffalo are subject to very severe movement restrictions and other restrictions in terms of being able to trade in animals and in meat products. And then also there is this continuous surveillance, because if there is spillover from buffalo, it's invariably going to happen in the communal cattle alongside the infected buffalo, and that is your first signal that there's something going on and you need to tighten things up a little bit. The African buffalo maintenance host, we have a lot of populations throughout South Africa. Kruger Park is the largest, and it is infected with all 3 serotypes. And these buffalo become infected for the first time once maternal antibodies have waned. So buffalo have synchronous breeding. At 3 to 6 months, you get waning of maternal antibodies, and the moment that happens, they will start becoming infected with whichever SAT types are circulating in that natal herd. So when you do the sequencing of these viruses from buffalo, it actually reflects the natal herd that they originate from. And we use this zoning method both in South Africa to control the disease. So around Kruger, we have buffer zones, we have inspection zones, and we've managed through all of these different processes really to contain it to the Kruger Park for a number of decades, and quite successfully. And other countries that also have FMD-free status, zonal status, use very much the same sort of approach, where you have a free area where you can trade, and those are indicated in white in Namibia as well as regionally. And then you have infected areas, and then you have areas where you have protection and there is surveillance and vaccination going on. Those zonal areas very much reflect the distribution of the African buffalo and the distribution of FMD-infected populations within the region. So if you look at this chapter, it's actually published in 2023 by Cornelius et al. They estimate that we have about 500,000 Cape buffalo on the continent, mainly concentrated within East and Southern Africa, and all of those buffalo populations are infected with the SAT types. So if you do a serial survey of buffalo and you do it at age 1, all of those buffalo, or about 90% of the buffalo, will be seropositive to SAT1, 2, and 3. They will establish an infection, and an individual buffalo can maintain that infection for a number of years. So you'll be able to recover the virus from throat scrapings of buffalo through repeat sampling. The rinderpest pandemic resulted in a huge reduction in African buffalo populations, and with that, very likely the disappearance of foot and mouth disease from the 2 southernmost buffalo populations. So that's Shishlu and Filosi and the Addo Elephant National Park. Kruger Park, though, did not undergo such a huge bottleneck, and the virus survived, and it continues to persist there today, and there are quite high levels of variation there. We have also, though, through successful breeding of foot and mouth disease-free buffalo, now have very large populations of African buffalo free of FMD outside of the historical endemic area. And this actually poses significant challenges in this time where we have FMD present in all 9 provinces. Because the last thing we want is for any of those FMD-free populations to become infected from cattle, and you have this reverse spillover. It has already occurred. So Shishlu and Filosi used to be FMD-free. It is no longer FMD-free. And the moment that happens to all of these scattered populations throughout the country, you then need to create these surveillance and control zones, and that is going to be incredibly expensive for the country. So we really do need to protect our buffalo, and especially the FMD-free buffalo. And you will see that based on statistics from 2023, the 75,000 registered disease-free buffalo are valued at around 16 million rand. So that's a substantial chunk of change. Another important component about the SAT types is that we have genetically as well as geographically distinct viruses. So when you do the sequencing of buffalo viruses, and these are from different game reserves throughout Southern Africa, from South Africa, Zimbabwe, Zambia, and our neighboring countries, what you will see is that they're clustered genetically in distinct groups, but also in geographically distinct groups. And we refer to this combination of genetic and geographical clustering as topotypes. So they are distinct within a specific topography. And you'll see that for each of the SAT types, whether it's SAT 1, 2, or 3, that at least for South Africa, we only have 1 topotype present within our borders. The same cannot be said for Zimbabwe, which has 3 different topotypes, and which also makes control of the disease through vaccination incredibly complicated. So the sequencing that we do to recover those topotypes actually is very useful for guiding how we design our vaccines and how we do vaccine matching. So historically, we have regularly sampled buffalo, regularly sequenced and typed the viruses, and then match those field strains, which will be the origin of spillover to cattle, to the vaccine strains. And we do that through phylogenies, but also if you look at the viruses antigenically, you can see that there's a link between this genetic and antigenic diversity. It's also really important to keep in mind that viruses usually have 2 distinct patterns when it comes to how they group within these evolutionary trees. They will either group temporally, like your flu viruses do, or they will group geographically. The moment they group geographically, things become a lot simpler from a vaccine point of view, because you only have to match to whichever topotypes or antigenic variants are present. So this antigenic variation mirrors the genetic variation, and because of that, we can use the typing to guide how we do vaccine strain selection and matching. And this is actually work that was done by Jan Esterhazen many years ago, but it is still applicable today. Nothing really has changed. And if we had to use those historical vaccine strains, they would still do a very good job because they match geographically what we have in South Africa. And we know if you take South African strains in your vaccine and match them to South African viruses that are circulating in the field, that your R1 value is above 0.3 generally. And that is an indication of a good match and that you're likely to have protection. If, however, you compare strains from South Africa with strains from Zimbabwe or Zambia or Namibia or Botswana, what you find is that the R1 value tends to be far lower, which suggests that it is not a good match, but we don't only go on the R1 value. So the potency of the vaccine is also really important and something to keep in mind in the current situation where we find ourselves. In terms of FMD outbreaks, it's important to understand how the virus is transmitted. And the main route of transmission is by inhalation of aerosolized particles, specifically for cattle. And usually you require close contact. So you don't have this long distance dispersal. It can happen under a very specific set of circumstances, but it is incredibly rare. If it were to happen, we refer to this as indirect or airborne transmission, and we consider this to be a low probability but a high consequence event. The reason for that is we control FMD through animal movement and by restricting movement and quarantine. If you have aerosol transmission, it is going to move even though those animals are not moving. And that is a whole different beast. Thankfully, it's not something that we've had to deal with, and most people don't believe that indirect or airborne transmission is anything that we need to be concerned about. I have already mentioned that the infectious dose needed to establish an infection varies depending on the livestock species. So for cattle and sheep, it is low, but for pigs, it is far, far higher. So the number of virus particles that a pig needs to breathe in to establish an infection is much higher, and generally, they do not establish infection through the aerosol route, but usually through ingestion of the virus, so through the oral route of infection. We can, however, and we've had a lot of really good experience with controlling the disease through quarantine and targeted vaccination. So previously, if we had an outbreak, it would be culling, very expensive, and also ethically becoming incredibly untenable. But if you do targeted vaccination and it's well matched, you can actually vaccinate to slaughter, and that is an approach that we use to great effect in South Africa, and it's been very successful in the past. It's very important, though, to remember that even if you have vaccinated your animals, the vaccine does not prevent infection. Just like COVID, if you get a COVID vaccine, it will not prevent you from becoming infected. What it does do is it prevents you from getting really, really ill. Okay, so sometimes people may not anticipate animals getting ill after they've been vaccinated, but it can happen. What is critically important is that if you've matched your vaccines and you have a good antibody response and you have neutralizing antibodies, is that vaccination will reduce the levels as well as the duration of viremia. And this is really important if we're thinking about controlled slaughter and also wanting to interrupt animal-to-animal transmission. So the moment you have really good vaccines with long-lived immunity, you're in a much better position to actually control the disease. And this is the strategy that we're employing now to control FMD and try and get on top of it again. All right, so I'm going to shift gears a little bit because we do have an FMD carrier concern. And the FMD carrier concern actually drives a lot of the international regulations which filter through to national level. So when we refer to an FMD carrier, and this is a state that was first described by Beckham in 1959, We define a carrier animal as one from which you can recover infectious foot and mouth disease virus more than 28 days after first infection. And this will differ. So carrier status can be established in a number of livestock species, up to 9 months in small ruminants, 3.5 years in cattle, and up to 5 years in your African buffalo. But it is dependent on strain, route of transmission, and there is a good deal of variation within that. The problem, though, from an international trade perspective, is that if you have carriers and you don't know that they are carriers, there is a potential for transmission if those animals are moved, either within a country or overseas with trading partners. So the carrier status conundrum, as we refer to it, really does drive a lot of the policy. But if you look at the investigations that have been conducted to date, we have not been able to prove that carrier cattle can transmit to naive in-contact cattle. We've also repeated the same process with African buffalo, and a number of those carrier buffalo were in contact with naive cattle and did not transmit the virus, but we had a few examples where it did happen, but not from carrier cattle to naive cattle. Yet the carrier worry actually drives policy to a huge extent. The carrier status, if we're thinking about vaccines, can be redefined, and it has recently been redefined as being the point at which an animal will clear an infection. If an animal has been vaccinated, it will clear it a lot faster. So it reduces the amount of time that it takes for an animal to recover from an infection. Which now brings me to some of the research that we've done, which actually deals with the fifth quarter and the risk that is posed by sending animals infected with foot and mouth disease that have recovered from the infection to slaughter and to controlled slaughter. Now, if we're sending animals for slaughter and they are in the subclinical stage, they are essentially within that very early time period, they are shedding virus, but you will not pick up any clinical signs of infection. That is a huge worry when you're getting into a system where those products might actually get back into pigs, for an example, and pigs are huge amplifier hosts. So that is what we worry about, and a lot of the regulations actually deal with that subclinical phase where you have virus shedding. And this is acknowledged if you look at the policies that guide slaughter regulations in South Africa. It's those early phases where there's no clinical signs of infection that we're really worrying about when we're sending animals for slaughter. However, we do need to stress that if you have had an outbreak, for example, in a feedlot like we had in the Heidelberg feedlot, 112,000 animals within a feedlot, and FMD swept through there, you now need to declare a day zero, which essentially means that those animals, every single one of them has recovered, or you have managed to vaccinate all of them, whichever of the two comes last. And then what you do is you have to wait 14 days from declaration of that day zero before you can send animals for controlled slaughter. So essentially, what that does mean is you're not sending viremic animals, either clinical or subclinical. You're sending fully recovered animals for slaughter. The problem now, though, is that some of those cattle will have become carriers, and we have no way of predicting what proportion of animals will become carriers. So the animals that are fully recovered pose no threat at all. The animals that have become carriers and have established a persistent infection will have residual amounts of foot and mouth disease in that oropharyngeal region. And that is where all of the disease risk mitigation measures are actually focused on. But keep in mind, we are sending recovered animals to slaughter. So what do these regulations look like, right? You've gone through an outbreak, you've been quarantined. If you're in a feedlot of that size and there's no vaccine, you have to wait for that virus to move through that feedlot. That can take weeks. If you do have vaccine, you can vaccinate, and then you can at least start doing controlled slaughter. You have to wait for the declaration of day 0 by the state veterinarian. Once that happens, then you can send your cattle for controlled slaughter at an FMD designated abattoir. So it can't just be any abattoir, it's going to be an FMD designated abattoir with a whole lot of security, biosecurity and risk mitigation measures that come with that in paperwork, etc. And then you have two choices. You can either send your animals for early slaughter, and that's day 15 to 41 post day 0, in which case there has to be carcass maturation and confirmation that when you test 24 hours after maturation, the pH has hit 6 or less. That's to ensure inactivation of the virus. If it does not, the carcass is condemned. You also, after maturation, need to debone the carcass and you need to remove all the major glands. That slows things down tremendously and you can send fewer animals can be processed. So you can imagine all the economic impacts that this actually has. And then there's also treatment and disposal of fifth quarter derivatives. So this includes tongue and offal, as well as the head, because the high-risk sites are there, and the feet. A lot of people who have undergone or experienced foot and mouth disease outbreaks, because of these regulatory requirements, are actually choosing not to send their animals for early slaughter. They just take too big a financial knock. And in fact, the abattoirs also, most of the money or their break even comes from heads and sale of that and offal. So some of the abattoirs have also gone under. Most people are choosing to send animals for late slaughter, so that's day 42 to 3 months, and that's been a recent revision in February. You still need to do your carcass maturation, but now you don't need to debone or degland, but you do need to treat head and feet. So either treatment or disposal. So there is still this cost and waste associated with that. So essentially, what we did when we designed this project is we asked, where is the risk? And keep in mind that you are sending recovered animals for slaughter. So you've defined your day 0, you've waited 14 days. The risk is really the carrier status. And you don't know which proportion of animals will become carriers. It ranges based on literature from 20% to 60%. So let's say, for argument's sake, you're at the 20% and you're sending all of those animals for slaughter and you're doing early slaughter, you're getting rid of 80% of perfectly good risk-free meat and protein. So we designed the project so that we would be sampling animals at the earliest possible time point when they are sent for slaughter, and that is at day 15. You do not know how many things need to align to be able to do this project. So you need to identify your feedlots. You have to have the FMD diagnosis done by the laboratory. You have to have vaccines to start the vaccination. You have to have an early declaration of day 0. Then you need to get all the Red Cross permits, veterinary approval, Section 20 approval, ethics approval, research approval. All within 14 days so that you can actually sample those animals. Our timelines for this was really, in reality, about 7 days to get everything in place. We tried it once, we missed the boat, we didn't get the Section 20 at the last hour, but we managed to get, thanks very much to Sean Morris, who is sitting at the back of the auditorium. He found the feedlot, we had this narrow time point, and we could actually investigate where the risk is. If we're looking at virus persistence in cattle and we're looking 28 days post-infection, which is exactly where the cattle will be if you've declared a day 0 and you've waited 14 days, then we know we're unlikely to find it in peripheral sites like lungs, interdigital cleft, because these are cleared relatively early, and so is blood. And because blood is cleared relatively early, also muscle tissue is safe, but we're still doing the pH requirement. If we're thinking about offal, then lungs and kidneys are your highest risk site. They take the longest to clear the virus. So when the virus comes into the animal, it basically disseminates through the entire animal. And then as the immune system kicks in, it starts clearing, and then it moves to the head and to the feet, with head being the high-risk site. If you are able to detect the virus within the head region 28 days post-infection, it is going to be present at very, very low levels. Yet we're still discarding of all the fifth quarter derivatives. So we had a look at the science to guide the design of the study. And there was a paper published by Pacheco in 2015. They primarily looked at serotypes A and O, but there were 4 carrier animals infected with SAT2 virus, some vaccinated, some not. And they sampled all these different head site regions to try and get some idea of where the virus persists. Now, what's very interesting if you look at those results is you will see all those color-coded sites in the head region correspond to where you will find virus in the SAT2 infected carrier animals. Far fewer sites than what they found for the European serotypes A and O. What's also really interesting is we're having to treat and discard the tongue, but there's no evidence of FMD in any of the animals that they evaluated, and also not in the lingual tonsil. So when we designed the study, we decided to sample all of the positive sites in the head region, so a large number of head sites. And then we also sampled major lymph nodes, red bone marrow, because that is where you expect the virus to replicate, as well as a couple of organ sites, with the idea of answering the question, where is the risk? We sampled 90 cattle at the earliest possible time point within the early slaughter phase, and we estimate, based on back tracing, that those animals were infected between 28 to 32 days before they went for slaughter. So this is the earliest possible time point we could have hoped for. On that slaughter line, we did 20 sample types, and they are all listed there. And we collected those in duplicates. They were either stored in DNA RNA shield or in buffered formalin, and both of those storages options inactivate the virus. And we needed to do that for the Section 20 requirements. The RNA extraction was an extensive process which essentially removes all of the DNA and only leaves you with very high quality DNA because you do a double cartridge purification as well as a DNase digestion. And then what's absolutely critical, if you think about the low levels of virus that we were expecting to find, is we did not pool our samples. We tested each and every sample individually. And this would only be possible with very generous support from RMIS and Zutas, and I'm immensely grateful, we all are, for the support. It was an expensive project, and we were hoping to establish a baseline. We were expecting and thinking perhaps we had sampled these animals far too early, and we were going to get a lot of positives, but you'll see that the results exceeded all expectations. So these were the lists of samples we collected. The animals came in for slaughter at the end of that day's regular slaughter. We had a number of people on the slaughter line, and we essentially worked through the night processing all of those samples. We then eventually, we collected the samples on the 25th of August. We were only able to test those samples in November because we could not get approval to test the samples. We could only get approval to collect, transfer to Hans Hoheisen, and store. But once we were finally able to get to those labs, the one thing we had to show is that those animals had been infected with foot and mouth disease. Now, the enrollment criteria for the cattle in the study was that they should show clinical signs of FMD infection. So it was 90 cattle that had signs of FMD, but we had to show that they had actually been exposed to a lytic phase and a replicating phase of the virus, and the way in which we do that is using NSP ELISA. And that's because our vaccines that we produce are inactivated vaccines, which are empty virus capsid cell, purified capsids. There is no genome within those purified capsids, so you will only get antibodies to the non-structural proteins if it's actually gone through an infectious phase and been exposed and gone through a lytic phase and circulated through the blood. So this NSP ELISA that we use, it's important to keep in mind that there is inter-individual variation in immune responses, and we use it as a population-level test. It's not an individual-level test. So the specificity is incredibly high. It will pick up all of the serotypes and FMD, but it is not particularly sensitive. The test that is incredibly sensitive is the PCR test, so the RT-qPCR test. And for the first phase, we used a commercial kit, very expensive, but worth every single cent. So within that triplex RT-PCR, you're picking up the virus. You're also detecting exogenous RNA, and this is a control that you add when you do the RNA extraction. That will give you an indication of whether you may have false positives due to inhibitory substances within your sample. We also target endogenous RNA. The endogenous RNA is an mRNA transcript that is produced in a wide variety of cattle tissue samples. And that gives you confirmation that the RNA extraction procedure worked really well and you have high quality RNA in your sample. And those textbook results that you see there on the right are actually from our study. They're not from a textbook. I could not have hoped for anything better. But I also have to say that this is the one project where I have never, ever prayed so hard for negative results. Most of the times I want my PCRs to work. All right, so in this first phase, just a quick snapshot of the results. Those cattle that were sampled on the 25th of August, 58.9%, nearly 59% of them had antibodies to your non-structural proteins. And that's pretty much on par with what you get for this test. What's really interesting is we sampled and tested a very high-risk site within the head region to try and get a handle on how many of the animals may have become or established a carrier infection or status. And it's sitting very close to the top end of 60%. 57.5% of the animals, we could detect FMD in that sample type. It was incredibly disappointing to get 3 lung samples positive for PCR, but the bone marrow was negative at the 35 cutoff, but also at the 40 cycle cutoff. It was just a flat line throughout. Absolutely no evidence of virus survival in the bone marrow. So this was really exciting and provided the first strong indication that the current deboning requirement is not necessary. If you had to assume that the levels of virus survival in bone marrow are as high as what they are in that high-risk head site, and you had to ask the question, what is the chance that we missed a positive bone marrow sample? It is close to 0. So the fact that so many of those animals actually had detectable virus makes that bone marrow negative result all the more compelling. It's also important to remember, though, that if you get a negative PCR result and you've included all the possible controls you can, which we did do, that a negative will confirm absence of the virus, but a positive PCR test does not necessarily mean that the virus you're picking up is infectious. And this is a really important distinction. So we had completed phase 1A, couldn't afford the commercial triplex PCR kit anymore, but we did rerun all of those samples with an in-house test, which had an exogenous as well as the FMD, so it was a duplex test. And it performed well. We got exactly the same results with those 3 sample types. So then we moved on to addressing the off-hole requirement by testing lung, kidney, and spleen. those three major, the lymph nodes, specifically mesenteric lymph node, which drains that region, and then the three major lymph node types, which drain different portions of the body, so hind, fore, as well as organs. And then also the tongue requirement. We had a look at that lingual tonsil as well. And this is just a snapshot of the results from this phase, comparing your NSP ELISA. You can see bone marrow was negative, spleen and kidney were negative, lung we had 3%, lingual tonsil, one of the animals came up positive, so that was also quite disappointing. And then if you look at mesenteric lymph node, this is an indication of whether or not you need to dispose of the offal, as well as those organ tests, 0%. Prescapular and inguinal lymph nodes, we had 17.8% and 3.4%. So the question now was, is the virus in these positive samples infectious, or are we just picking up small remnants of the genome as the immune system is busy mopping up and clearing the virus from the system? So let's go back to the biology and what we know about the virus and its replication. To be infectious, you need to have an intact genome. So you've got to have a very large intact RNA fragment. When we do a diagnostic PCR, we are only targeting 108 of those 8,300 nucleotide genome region. It's a tiny, tiny portion of the genome. So what we did is a quite extensive experiment to look at viral genome integrity. And we essentially tried amplifying different fragments of the viral genome, starting with your 108, and all of those were positive. These are the positive samples from this first phase, and then moving up to bigger and bigger fragments. And you can see that as you move up, you detect fewer and fewer positive samples. And actually, once you get above 500, everything is negative. We also included endogenous controls that target mRNA transcripts in the cattle genome, and we had two there, one of 289 base pairs and another of 629 base pairs. All of those were positive. What this essentially means is that the negative results we got for the FMD were not due to RNA integrity problems. The RNA quality was really, really good. But there's no infectious FMD virus in any of those samples. So this was really, really significant. Which then brings us back to the regulations that we are using and which are guarded by WOHA and the terrestrial manual, and the publications that they are based on. And they are based primarily on this Cottrell paper. So the big scare with bone marrow is, in fact, that the virus can survive in chilled bone marrow for up to 4 months and frozen bone marrow for up to 7 months. But if you go back to the original paper, this was from viremic animals that were sent to slaughter, not from recovered animals. So the regulations that we are using are not keeping pace with the additional risk mitigation measures that we have put in place to make sure that we are only sending recovered animals to slaughter. And in fact, if you are sending animals from FMD-infected premises where a day zero has been declared, you can have far more confidence in the FMD status being fully recovered than what you can for animals that might not come from infected premises. And yet we apply these really draconian regulations. If you go back to the Cottrell paper, quality is not that great, but look at the blood results. You can't pick up FMD in blood 6 days post-inoculation. Kidneys are cleared at 8 days, spleen at 6 days, liver 6 days, bone marrow from 8 days. So actually the results we got with the SAT2 type perfectly match what was published back in 1969 and what is used to guide the current regulations. So based on this, we have proposed to the Directorate that the foot and mouth disease controlled slaughter regulations should be changed as soon as possible. We don't need to have a late and an early slaughter. You only need one controlled slaughter phase from day 16, that is when we sampled, it can't be day 15, up until 3 months post day 0. You still need to do carcass maturation, that's a standard practice, but I'm not sure that we need to get to a pH of 6, because that is to basically deal with viremic animals. These are recovered animals. You don't need to debone, you don't need to degland the carcass, you don't need to dispose of offal, and you don't need to treat or dispose of the tongue. For now, because we haven't completed testing all of those samples within the head region, we are not going to recommend dropping treatment or disposal of head and feet. And we anticipate that when we get those results back, we will be picking up some infectious virus, because that corresponds to what's been published in literature. But there are ways of dealing with specified risk material on the slaughter line. And Lisa Oerdendahl, who's also sitting here, has tested this out on slaughter lines. You can remove the specified risk material. There's no need to discard the entire head, but that will be the next phase. So this recommendation is based on science on SAT2 types to guide outbreaks caused by SAT2 types. And unfortunately, if you look at the literature, current policies conflate virus survivals and risk of animals, viremic animals sent for slaughter, with those that are recovered. We need to acknowledge we are sending recovered animals for slaughter if you're doing controlled slaughter. In short, the disease mitigation measures and risk mitigation measures that we are putting in place are doing the job, they're working. The risk has been removed from all of those parts of the cattle. Right, so I'm going to quickly, within the last few minutes, just speak about the current situation and the deterioration or the apparent deterioration of FMD control in Southern Africa. And I've actually taken this from one of Gavin Thompson's slides. He presented this at a conference in Botswana in 2018. And what the graphs show, and we should take care not to oversimplify, as he has pointed out, is that at some point we were doing a really good job with zonal FMD-free status by using a combination of methods, biosecurity, vaccination, etc. What's really interesting is that the moment we introduced vaccination, you see this dip for 3 SADC regions, and the moment we started producing our own local vaccines that are matched to outbreak strains, you see another dip. We maintained that for a while, but unfortunately in 2006 we stopped producing our own vaccines, and now you see this increase in outbreaks taking off there. Unfortunately, that graph of Gavin's only went up to 2010, but if you are interested in what it looks like now, we've jumped up a couple of levels, and it's pretty much off the charts. So we now have FMD in all 9 provinces, and in 2025, you have this enormous jump in the number of cases that are being reported. And you can see all the color coding there, 2025 in orange, 2026 in red. We're in a really, really dire state at the moment. It's not just us, though. So we have SAT viruses that have gone out of Africa into a number of other countries, and we now have concerns, and it may be applicable to us, because this SAT1 virus is on the move, and we have SAT1 outbreaks ongoing in South Africa, that vaccine strain or vaccine availability for SAT1 might become a problem again. We've just managed to come out of vaccine shortages. So this is something we definitely need to keep our eyes on. Also, just a few lessons from the past. If we look at 2 serotype O outbreaks, one that happened in South Africa in 2000. So this was a virus that doesn't occur in South Africa, introduced through swill feeding to pigs. Remember, I said the ingestion is one of the routes of establishing an infection in pigs. And what we worry about with pigs is these high levels of virus amplification. So one pig can produce what 100 to 1,000 cattle can produce in terms of virus aerosolization. Not good if it gets into pigs, and unfortunately at the moment FMD is in pigs. But have a look at the commonalities between the two. Unfortunately, in South Africa, the people in KZN had not dealt with foot and mouth disease outbreaks. So they missed the initial one that happened early in September. But then it moved and it was caught and it was rapidly detected and typed. Here in South Africa, within 24 hours, serotype that doesn't naturally occur here. We didn't have vaccine available, but very, very strict biosecurity was imposed. And in fact, the army was brought in and there was culling, very similar to how the Germans handled a recent outbreak that happened in water buffalo. A no-holds-barred approach. Detect quickly, take drastic, immediate action. They managed to identify a containment zone, so it didn't affect the entire country, and they've just had their disease-free status reinstated. It took a bit longer in the South African situation because we didn't get it right away, but if you do, your success of containing is much, much better. And this is a really tough lesson. FMD is brutal if you are complacent. You have to move quickly. You have to pick it up quickly. You have to deal with it very, very quickly. We are on a very, very long road to recovery, and that first step in recovery is vaccination. Everything hinges on vaccination. We can't throw the rest out. We still need all the biosecurity and all the other measures that we use, but really to get back on top of things and start reclaiming FMD-free zones with vaccination, we are going to need millions and millions of doses of vaccine for our 24 million herd of cattle. It's also jumped across into pigs, which is a problem. But I think what we have learned is that targeted studies on our serotypes should guide the policies we're going to use on our serotypes. And we're going to be undertaking a 5th quarter research project on pigs. Pigs do not become carriers, yet the slaughter regulations that are being applied are those that are applied for carrier cattle sent to slaughter. We will need expanded laboratory capacity. At this stage, we have one laboratory doing all of the diagnostics, and they are completely swamped. The moment you start vaccinating, you need to do post-vaccination monitoring. So there are going to be millions and millions more samples coming in that need to be evaluated. We need additional laboratories, and those are actually being assessed and will be added so that we can start building capacity there. We also really do need to protect our FMD-free buffalo from a reverse spillover from cattle. If we don't do this, we are going to be in a really, really difficult situation. The best way of doing that, because they are such efficient maintenance hosts, is to actually vaccinate the cattle around the buffalo so that it doesn't get into the buffalo population in the first place. Also just want to say that there are unfortunately many, many, many people that have been affected by FMD. The situation is dire, it's absolutely unprecedented. But farmers on the ground are actually our eyes and ears on the ground. And I would really encourage everyone to record, observe, sample, and store, so that we can at least learn from today's disaster and prevent a repeat happening tomorrow. We really need all hands on deck. I also just want to mention this is a transboundary animal disease, and that means that we actually need cross-border diplomacy. I'm not sure that we are ever going to get to a point where we are dealing effectively with this disease if we are only worrying about what's happening within our borders. We have neighbors alongside us that have vaccine stocks that they may not need. And the virus is getting closer and closer to the borders. I know it may sound very altruistic, but why not give that to South Africa and at least protect your borders on the other side and retain your FMD-free status? We should be thinking about regional vaccine supply stocks and how best to direct them so that we can keep as many zones free of the disease as possible. learn from the successes of others. I've just given you 2 brief examples of the South African and German situation. Rapid detection, rapid response, decisive action to contain and eliminate the virus. If you miss it in the early phase, it's almost impossible to catch up afterwards. And then lastly, I just want to emphasize the importance of public-private partnerships. This thing is bigger than all of us. There is no ways that state can control it or the private sector can control it. The only way we're going to get ahead of it is if we actually form effective partnerships that allow us to be agile, have a rapid response, as well as a decisive response. And I think the 5th Quarter Research Project is an example of this working in practice. It all began actually with industry going to government and questioning the 5th Quarter regulations that are in place. Government then went to academia and to Prof. Naidu and asked for a recommendation on the 5th Quarter regulations that apply, nationally as well as internationally. And Melvin Kwan, who's also here, and Mary Lou, who's online, and I worked on a recommendation. And to be honest, I worked with Gavin for many years, Gavin Thompson, the late Gavin Thompson, and he was very, very passionate about how dreadful these regulations are and about commodity-based trade. But I didn't completely understand it until I finished this regulation. And I realized we cannot leave it here. We are applying things using science or guidelines that are for serotypes that don't occur in this country. And so I reached out to Sean Morris and to Dirk Verwoerd, and we shortly after that we had a meeting. Lisa, who mentioned, I know now more about slaughter regulations and what happens on the slaughter line than I ever thought I would want to know. But a lot of people came on board, and within a very short period of time, by the end of July, we had a Section 20 application ready, and we just missed getting approval for that. After finally getting those samples and testing it, there were so many people that came on board. I don't have enough space. I probably drew many, many slides of all the people who've willingly and freely gave advice. We need everybody on board. We all come with our own set of expertise. To be able to fight this properly, we need to pool those resources. And you can only do that if you have everybody on board and understanding just how dire the situation is and that we need to move forward. So these are just some of the people that I really want to thank. On the slaughter line, in one night, we collected 1,800 samples. There were 13 people on that slaughter line. They didn't really slow it down. They slowed it down a bit, but not that much. But we collected everything, and obviously you have to have veterinarians who know a lot about head anatomy and how to collect those sites. And thankfully we had a lot of them and experienced veterinarians on board. This wasn't their first rodeo. We also had a sample processing team which started with 6 of us and then got bumped up to 11 and worked through the night. And a special thank you to Nicolese for the sample box design, which Vanessa and Erin are busy holding up there. That saved the day. We were going to do it in individual tubes. I don't know, we would never. We would have probably taken 24 hours to do the project, the sample processing. It was very frustrating having to collect, transfer all of these samples to HH and then sit and wait for approvals. But we are resilient, we are persistent, we know the procedures now, and I'm sure it's going to go a lot faster. And how lucky are we at the University of Pretoria to actually have a facility that is approved for FMD research. There are only 2 facilities, aside from the TAD lab, where you can do the diagnostics, where you can actually do FMD research. And that's because it's in the FMD endemic area. We had those samples in storage up until the beginning of November. To date, we have extracted RNA from more than 1,400 samples, or I should say the 2 ladies sitting there did most of that. We have tested all of those samples with a diagnostic PCR. I've run close to 3,000 PCRs. We segregated all the duties, and a lot of sequencing of the positive samples has been done, all in the space of 8 months. I know to people outside South Africa that may seem like a really long time, but for those of us dealing with Section 20 research and animal ethics approval, I think we've done pretty darn well. And I have many, many thank yous to say to a lot of people, some here, some not. Staff at HH who keep that core facility and satellite campus going, 2 deans from 2 different faculties who made it possible to get research assistance for the project as well as equipment, and the entire 5th quarter team, both here and online. Financial support would not have been possible. This is a new benchmark in FMD research. It really is setting a precedent in terms of sample numbers, number of animals sampled, and it's on an SAT type, which is underrepresented in literature. And I also want to thank my ministerial task team colleagues that are here in support. I appreciate all of the many hours we have spent in online meetings having insightful and sometimes challenging discussions on FMD. I've learned a huge amount from having experts concentrated in one room. But we are also very lucky to have experts across this country and be able to galvanize all of that expertise. Vanessa and Erin were my right and left hands, would not have been able to do it without them. They were vegetarians before the start of the project, and that status remains unchanged. Thank you very much.
Let's give you another round of applause. I must say this was truly instructive and reminded me of the expert lecture on COVID-19 when I saw all those sequencing and things. And knowing how measured and rigorous Professor Bastos is, it is no surprise. And we are now going to get into the question and answer session. And if we just keep to the ground rules a bit, we'll open for questions. So you may ask 1 question, maybe 2 at most per person. Because if there are too many questions in a sentence, you know that's what politicians do, Minister. Many questions in one sentence, and you just answer one, they don't even remember that you didn't answer the rest. So let's keep it, in such a way she can be able to give effective responses. And also, part of the ground rules, let's direct the questions to the expert lecture. It's quite a very emotive subject. It can get us very angry and excited and emotional. Not to the minister. Because I know it could get like that. So let's just keep it within the expert lecture. He will ask questions if he has any. And I'm sure he'll take some of the notes to the president, his boss, who keep Buffalo, yeah. So that they can learn a bit more about what's happening. So, ladies and gentlemen, I'm opening this floor now for questions. 1 question, 2. Minister, number 1. Please go ahead.
Thank you very much, Professor Bastos, for this. It's also been a very exciting set of research done. Thank you. It's been a very exciting set of research done, and we've obviously been waiting for it so that we can start to look at how we can adjust the regulations led by science to be able to take this into consideration. The one question I have, which nobody's been able to answer to date, and it was from the first part of your presentation, is that if one looks at all of the literature and academic research, the types O and A, and the European strains particularly, have been the ones that have generally presented with very strong clinical symptoms. Yet what we've experienced in this current outbreak with SAT1, 2, and 3 is very, very strong clinical symptoms, quite more severe than we've probably ever seen before. And I wanted to try and understand for you why that is the case and why we've seen this change that has not been present before. Thank you.
Thank you, Minister. Go ahead, Prof. Yeah, come here if it's not.
All right. Is it on? Hello? Okay. Yeah, that's a really interesting question, and I think it actually has come up at the FMD in Durban, but I don't think you were there for the tail end of it. The viruses at the moment, the SAT types, we do usually see that it is a subclinical or mild infection. But there are strains that have caused clinical infection in buffalo. So it's not that we can say that all of them will always be subclinical. But I think the way that the viruses are behaving now, they're behaving very much like livestock viruses. So very much like the European serotype viruses. And I think that this is potentially because they have been in livestock for quite some time now. And it is really a livestock cycle that is driving things at the moment. But it's speculation, so.
Thank you. Another question?
This is a stand.
There's one here, the lady in the middle of the room. There's a mic coming over there next to you.
Bring the mic.
Where are the mic people?
Thank you
Students,
for the presentation. I just have one quick question. So we know that this outbreak sort of got out of the main area in 2019, and we started seeing an acceleration in the last maybe 2 years, so to say. What is it that we were doing between 2019 until we got to, and then why did it suddenly start accelerating?
I thought I might have two gentle questions to start off with, but clearly not. I wish I could answer that. I think it's probably a combination of failures on multiple fronts. Movement of infected animals, we know that that is a major issue. Also animals being taken to auction where you sometimes don't know that they are infected. So the way the virus moves really effectively is infected animals moving and transmitting. If you're really thinking about slaughter or meat products, the risk between a live animal shedding virus and meat products from it's nice and day. Okay, I'm gonna.
There's a hand there next to Lynette. He has a mic.
Okay, thank you very much for the interesting, presentation. I learned, about two things from your presentation, which I can take home. The first one is rapid detection, and the second one was to learn, from this particular, experience. Now, my question is: are we not able to predict, the outbreak? Now, the next one, do we not have, such, a capacity? Now, my understanding is, of course, the detection need to be, guided. Perhaps, some sort of, modeling, exercise and other things. And I think we do those type of, testing, for confirmation purpose. Because what I'm saying is that, as we had, some sort of, discussion, there are people who say that we knew that FMD is, coming to South Africa. So which means, my question is, do we not have, those capacity, for example, to detect, the next, outbreak?
Okay, so I think if I'm understanding correctly, it's preparing and bringing in a predictive modeling I'm not sure that that's really happening at the moment. I'm not sure how successful it would be. FMD is quite an interesting virus. It can fly under the radar quite a bit. But I do think if you had enough information, and that's why I say farmers being citizen scientists would be really valuable. If you could track the movement and actually start putting that into models, you may get to a point where it becomes predictive. But I'm not aware of any predictive modeling happening at this stage, but it would certainly be useful.
Thank you, Prof. There's a hand there. Yes.
Amanda, thank you very much for this presentation. It was not only excellent, but the work was also excellent. So one of the things, the question I want to come with is, within the MTT, we have discussed the possibility that this work was done with only one serotype. And then also from the buffalo work that I've been involved in over the years, is that a SAT1 serotype act a little bit different within these. Do you think we have enough information from all the A and O and also the SAT2 to make a generalized comment, or do you think there's a possibility that something like SAT1 might react differently within these animals?
So thanks for the question, Francois. I think we have a lot of information, and actually Prof. Francois Maria did a similar study on an S83 with Sean Morris a few years ago, which did change regulation and policy. So we have S83 information, we've got the S82, we've got the other serotypes. I know that there is concern about us using a blanket approach, but if you go back to the literature, go back to the Cottrell paper, it's there. We're actually confirming with SAT types what's been in the literature already for a while. So I actually feel quite confident in saying we've got enough information, let's change the regulations.
Thank you, Prof. There's another hand here on my left.
Thank you, Prof. I think it was an excellent presentation and great work done. And also Sean, where's Sean? I think he's a big hero. I also sense, obviously, the frustration in your presentation regarding regulations. And in my background, I'm from a different department, on Graduate School of Technology Management, and looking after project management. So when this started happening, we also did some work and find out what can we do, what must be done in order to vaccinate all the animals in South Africa within 1 year. So what is constraining us in doing that? First of all, availability of vaccines, and obviously regulations. So, and I think the Minister did a great job in getting the MTT and the ICC on board. I think that was a great initiative together with the government. But still, we are down the line. We don't have a Section 10, we don't have a Section 9. It's just going backwards and forwards. So my request, and maybe you can tell me how you can do it, is can't Dewald and these guys, no, and the MTT and the government, please lock yourselves in a room and don't come out until you've got a properly Section 10 addressing the regulations that needs to change, because we're in dire need of this. I will go on my knees and cry, because I'm also, my family's got cattle, we've been hit with FMD, I see abattoirs going bankrupt. We cannot just sit there and send emails to each other anymore. We cannot send out posts. This week, Section 10 will be signed, and then it doesn't get signed. I send it to our groups, we're all excited, and then boom, everything falls flat. So please, I ask you, and I will go on my knees, clear your diaries, get in a room and talk to each other, and please change these regulations. It's scientifically proven. It is archaic. We need this to change, and I will make myself available. We've run Microsoft project programs, schedules of how to do it, even with communal cattle, you know, using auction mobile kraals to get them together.
Thank you.
It can be done. Thank you.
Can you pass the mic on here? I'm sure officials are listening. Officials must just be together. And
Yeah. Amanda, thank you very much for a very good presentation. I've learned a few things about presentation skills. I was just curious about the proportions that become carriers in the different serotypes, whether there's any data on that. Thank you.
So that's the interesting thing, is we have no way to predict. It seems to be strain dependent. So, and that's the problem, because if you had some way of predicting how many carriers you have, or you had some way of detecting the carriers, you could actually deal with the worry about the carrier status. But there isn't a way at this stage of predicting. But I do think that if we do enough of these studies, we'll get to a point where we'll be able to start understanding more about what it is that may lead to establishment of carrier status. The other interesting thing is that the vaccination status doesn't change the proportion of animals that become carriers. So whether they're vaccinated or unvaccinated, it's the same proportion of animals for a particular strain that will become carriers.
Thank you very much.
So there was actually... I think I have to hand the mic over because there's good news, apparently.
Oh,
Section 10 was signed yesterday, and it's being gazetted this week, so it's done.
That's nice. Should we clap? Well done, Minister.
Well,
Appreciate that.
Thank you.
Prof.
Great job. My question is, I'm trying to put myself also in the position of the director, animal health, who's supposed to help the minister in changing the regulations. Has there been questions coming up of repeatability of the research in terms of whether it will produce the same results? That's one. And just to use the opportunity, I want to know if are you also on the scientific committee of WOA? Because it took years for WOA to accept commodity-based trade. And I see this as an opportunity with the outbreaks all over now to influence international standards. Thank you.
All right, so I'll answer the first question, the last question first. I think the change will probably come a lot faster. I think that the work is sound, and what we're focusing on now is it's a two-pronged approach. We are interacting with the directorate, and we've explained all the research, but we are also preparing a publication for this first phase. Because once it's gone through a peer review process, it can't be ignored. And it will be tabled, so there's an opportunity to table in May, and then I think there's a meeting in September amongst all the member states, the 183. So we are thinking about both national as well as international. Yeah, and then in terms of repeatability, it has come up, and Francois did allude to it. I'm confident that if we had to repeat this with different serotypes, that we're going to recover exactly the same results, even if they're slightly different in terms of virulence, pathogenicity, or serotype. But it would be good to get additional information, obviously.
Thank you, Prof. I'm going to allow 3 more questions. Actually, the third one is an extra question because I'm being nice. And then we can take some of the questions outside the hub there with her. Even if you make a note to have dinner, she can answer you. But we have to restrict the time now. There's one hand there, also students. I also want to hear from students. Let's start here, and then we'll go there, that side. Yeah.
Thank you. Thank you very much. Thank you very much, Dr. Zenon Ndaisenga from the Department of Political Science. I think my question is about the vaccination will cost a lot of money. And what if South Africa put a lot of money in vaccination, and the neighboring countries are not doing the same for vaccination. Don't you see any uncertainty about this? That is my question. Thank you.
Yeah, I think most countries want to avoid vaccination because if you have zonal FMD-free status without vaccination, that is like the top tier that you want to be in terms of trade internationally. But I do think that the moment it gets to a situation like we have in South Africa, where you're not able to control it through biosecurity and other measures, and you don't get it early enough, the only option you have is vaccination. And I think a lot of our neighboring countries have already thought this through. So I know Namibia in February last year invested huge amounts of money in placing an order for BVI vaccine so that they would be prepared for the eventuality of FMD spreading. So I don't think there is vaccine hesitancy initially, but once it gets past a certain point, we are very grateful for the vaccines.
Thank you very much, Prof. I don't know if we have the money. Maybe we do, Minister. It's another story. Who's a student? You look like a student. Yeah. Awesome.
Indeed, I am a student. Thank you, Prof, for your presentation. It was very wonderful. I just want to ask this, can you also maybe look at speeding up the processes of approvals when it comes to scientific research, like Section 20 and ethical clearance? I know you know I'm looking like that. Because, I mean, it's very ironical that I'm allowed to carry meat from KZN after a traditional ceremony back to Gauteng. But I cannot carry a small sample of my research when, so.
Do you really want me to answer that, Mkhalisi? I don't.
Yeah, he's bringing your crabs
Over a cup of coffee next week.
Yeah.
Okay.
Okay, there's the gentleman there.
Yes, thanks, Prof. I'm Themba Skakani, Chief Director of Veterinary Services, KZN. It's more of a comment on my side rather than a question. We all get entangled up on the behavior of the virus and all of those things, but we forget the one key aspect is the compliance of the affected communities. I mean, the outbreak in 2012, it talked to a sale of animals from Limpopo because people were hungry. The current outbreak around Mfolozi, because that's where it comes from, we did not have mechanisms to assist those communities to dispose of the animals and make a living out of them. So people in the house must understand that once you've catched the outbreak and implement a vaccination program, people still have to live. And if we can find ways and mechanisms of those people to get, I'm seeing this now with commercial white farmers. If you find mechanisms and ways, and that's why this excites me, if you find mechanisms and ways and understand within that those guidelines and the protocols and everything that people have to live, it always works. It worked in 2012, we got rid of the problem. This time we struggled to find mechanisms and reports of our contingency plan. The last one, it's more of a comment again, I'm happy to understand that there is a probability that we can eliminate FMD in the buffaloes in Mfolozi Game Reserve if we were to run a breeding program and removing the calves. What's your comment on that one? Thank you.
Thank you.
Good. Comment? You don't have a comment.
No, I think, yeah, it's a challenging situation. Compliance is, I think, always a problem. I mean, we have all the paperwork, the regulations in place, but it's actually the realities on the ground of dealing with outbreaks is very, very different. Yeah, and the buffalo, we have to keep an eye on that. It's an important component. I think vaccination is great, but in terms of where we direct the vaccines, it's got to be a bit longer term as well. Make sure we don't end up in a pickle, or a bigger pickle. Yeah.
Yeah. That was the last one, but there's a student, allow it.
All right. Thank you, Professor, for the presentation. My question is, what type of cattle breeds did you use in your experiments? Because if they were European breeds, how would they compare to our African breeds in terms of the expression of the virus or the immunity?
So it's not, I don't think it will be all that different. It is obviously something that I think we would like to look into. There are differences in literature, but as I've explained to other people who've raised concerns about differences between strains or breeds, we know a lot about this virus, and there's certain bounds that will apply to all strains, all serotypes, and all breeds. So there will be some variation of that, I'm pretty sure, but I don't think it's going to be so big that it would invalidate results, or that we're going to get anything really, really different. I hope I'm never proven wrong.
Thank you very much. Let's give another round of applause. Ladies and gentlemen, it remains for me to thank all of you for honoring us with your presence. And before I conclude, as a retiree, I have to polish the shoes of my boss. I've been a boss here for some time, and then he came and took over and pushed me on the side. So if you have the last word question or whatever, Vice Chancellor, this is your time. You don't have any, okay? You are witnesses. I polished his shoes. He said, I'm fine. I've spotted two deans here in the room. Thank you very much. Dean of Natural and Agricultural Sciences there, Prof. Maharaj and Prof. Stein, from our engineering, build, and IT. And all the other experts here, lecture students, our marketing team, for organizing this with the faculty and your support and the center. It is indeed one of those proud moments for us to have listened to this expert lecture. It goes down in our archives as one of the best that we have had. So we'll now break. The hub is, as you go out here, for those of you who have not been here, then there will be some refreshments served there. And we keep animals here on this campus, our experimental farm used to be. So we miss the beauty of the campus because they closed it. So we have to go there by spots. But thank you very much. We'll now break, and you can catch Prof. Bastos outside. And please don't make it difficult for her. She still has to go home and do more research. And to you, Minister, thank you very much for making time. It's not very easy to have ministers attend. So we have hosted the number here, but the record number I've seen is going to an event. We were speaking Lesotho, and there were 8 ministers. I'm like, Jesus Christ, it never happens where I come from. But thank you very much for taking time. You have heard for yourself, and I'm sure you learned a lot yourself through this crisis that you are managing. And let's now break for refreshments and thank Prof. Bastos again with the family. Had it not been for the support of your husband and children, it would be very difficult, we know, but I want to thank them as well. Martin is somewhere here, and the kids for supporting you throughout. Keep on producing this research. Minister, as you can see, we need more funding for research. And thank you. We have a gent now. Good evening. It was great.